I'm Leonardo Bonetti, Associate Professor at the Center for Music in the Brain at Aarhus University and Research Fellow at the Center for Eudaimonia and Human Flourishing at the University of Oxford. And I thank you very much for inviting me to give this keynote lecture for the 8th Annual World Music Conference. I'm going to talk about the neurophysiology of music recognition and on the impact that aging has on these processes. I will focus on two of our latest studies, but before, I'd like to introduce my presentation by asking this question: why memory, music, and the brain? So in other words, why do we do what we do? So I think that investigating memory is essential to understand the healthy human brain. And understanding a healthy human brain is very important, is a sort of first key step to investigate what happens when there is neural and cognitive decline, like the one associated with aging, and thus also studying what happens in pathological aging and potentially trying to use this information to develop clinical application for memory disorders. And the reason why we use music is because music is particularly good to study complex cognitive processes, such as memory, especially in the conscious domain. And this is because music is organized in hierarchical sequences of sounds. To some extent is a universal language, or at least is more universal than common languages that people speak, like English or Italian or Danish. And this is a bit more technical, but it also elicits a very good brain signal, especially when measuring that with MEG or EG. So with that, I will go straight to the first study and to generally how we collect this data. So in the first of the two studies that I will show you, we recruited 83 participants, which is a rather big sample for cognitive neuroscience. And as you can see, in this case, we started by studying healthy young participants. And we recorded the brain activity using two instruments, two machines. One is the magnetoencephalography, or MEG, this one here in this cartoon. And the other one is the magnetic resonance imaging, or MRI, that we use to collect structural data of the brain of participants. Now, I won't go into details here, but what is important to know is that when combined together, MEG and structural MRI, we can measure the brain signal with excellent temporal resolution and also with reasonably good spatial resolution. And in our studies, we mostly have an experimental design focused on first an encoding phase, where people are asked to memorize something, and then a recognition phase, where people are asked to instead recognize either something that they previously memorized or something novel. And today I'm going to focus on the recognition phase, because this is the data that we collected about. But to give you a better sense of how the experiment worked, I will also spend a few words on the encoding phase. So first of all, participants arrived in our lab, and the first thing that we did with them was asking them to listen to this music, to this short musical piece twice, and just try to memorize it as much as possible. Now it lasts about 20 seconds, and I will just play that for you to give you a sense of how it sounded. So once again, participants listened to that twice, trying to memorize this musical prelude as much as possible. And after that, we had the recognition phase, where the participants were exposed to a series of musical sequences in a random order, and they were asked to state whether they were previously memorized musical sequences or novel musical sequences. And what does it mean? That means that the previously memorized musical sequences were melodies that were taken from the musical piece that participants previously memorized, while the novel ones were variations, systematic variations of such sequences, variations that were built in this way, either changing all the four tones after the first one, which as you can see is the same as the previously memorized, or changing only the last three, or the last two, or the last one. And here an example of how they sounded. First, an example of the memorized musical sequence, and then an example of variation number 1, or 2, 3, and then 4. With that in mind, I'm going straight to the key results that we got here. So here we identify some of the essential brain regions that played a very important role for this task. And here you can see, for instance, the left auditory cortex, a rather large portion around the left auditory cortex of the brain, depicted in this brain template. And then you also got this 5-time series, one for each of the experimental conditions that we had. So in red, the previously memorized condition, and then in the shades of blue and black, the variations or novel sequences. And here we also got these musical sketches, the sketches of these musical notes to show the onset of each of the tones. And essentially, what we got here was like 2 different processes happening almost at the same time. So when we introduced the variation, for instance, at tone number 2, as you can see from this graph here, we got this rather sharp negative signal. And this is something that happened systematically whenever we introduced one variation at different points in time. And this is a sort of prediction error that is happening in the brain when the brain spots that there's something that is deviating from what was expected. And the other thing that we are seeing is that after introducing the variation, we don't only get this negative peak, but also we don't get this positive peak up here. And this peak stays only for the memorized condition at the end, suggesting that this is a process where there is a sort of match between the memory trace that was learned before and the incoming sound. Interestingly, when we move, still being in the left hemisphere, towards brain regions that are mostly associated with memory and evaluative processes, like hippocampal regions, parahippocampal gyrus, inferior temporal cortex, orbitofrontal cortex, we observe something similar, but perhaps even more refined. So when we have the variation inserted here, for instance, then we got this negative peak once again, and this happens systematically for each tone that we vary. And then at the same time, after introducing the variation, we don't get anymore this positive peak, as you can see here. And then, as a matter of fact, at the very end, the only positive peak that we see is the one of the previously memorized condition, reinforcing this idea that this positive component of the signal up here is showing the match between the memory trace and the incoming sound. Something else that is very relevant, although not very easy to see perhaps from these graphs, is that these negative peaks and also the positive peaks up here are happening in a similar fashion compared to the left auditory cortex, but they are also happening shifted in time of about 100 milliseconds, suggesting that there is a natural information flow from the auditory cortex to these higher order brain regions in relation to memory. And this suggestion was actually verified by using dynamic causal modeling and testing on a quantitative level the information flow between these 2 brain regions. Something similar happened also in the right hemisphere, although now for the sake of time I won't go too much into details for that. But also here you can't really see very much the negative peaks, but you can see very clearly the positive peaks up here with the same behavior that I described before. And that is even more clear when moving to the right hippocampal regions with very clearly these negative prediction errors, negative peaks for the prediction errors here on the bottom, and then on the top these positive peaks. With even some extra situations where, for instance, when we introduced the variations at tone number 4 and 5, we also got this positive peak of the prediction error, which is something very interesting that we will study more in the future. So the question here is, why is this relevant? And well, obviously it's relevant for the sake of basic research, because we describe the brain spatiotemporal dynamics of recognition of previously learned music, and also of the conscious prediction error that was emerging after we introduced the variations in this music. And beyond music, that's also relevant because we gain very relevant insights into how the brain processes information over time. So that's definitely relevant for basic research. But beyond that, we also got something more. So we got a sort of solid baseline of the healthy human brain that we can use to better study memory decline and neural decline in aging, both in healthy aging and then also in pathological aging, like in clinical populations, for instance, affected by Alzheimer's disease and dementia. And along this line, we actually then recruited additional participants, 40 older adults, with a mean age of 67 years, and everybody was older than 60 years old. And then we had 37 additional young participants, younger than the ones that I showed you before, with a mean age of almost 22 years, and everybody was younger than 25 years old. And once again, we used exactly the same machines, so MEG and structural MRI, and we also used the same experimental paradigm as before. And here we are with the results of these second studies. So in this case, similarly to before, I'm showing you one of the same brain regions that I mentioned before. In this case, we have these hippocampal regions in the right hemisphere of the brain. And we had that for 3 different conditions only, and not 5 in this case. So the previously memorized condition, as you can see here, and then 2 categories of the novel condition. The novelty 1, where we introduce the variation at tone number 2, and then we change all the sounds, and then novelty 3, where we change only the last 2 sounds. And in this case, you can see that we have two different time series. In red, the young participants, and in blue, the older participants. And what you can see here, perhaps, is that you can actually recognize these positive peaks happening after each sound when there was this match between the memory trace and the incoming sound in the memorized condition. And what is very interesting is that this peak is essentially almost always stronger for the young compared to the older participants. Similarly, when we introduce the variation, we get these negative peaks indexing the prediction error, showing this mismatch between the expectation and what actually is coming in terms of music. And as you can see, these negative peaks are much stronger for the young participants compared to the older adults, all over the place. And this is happening again systematically when we introduce the variation only at tone number 4. And in this case, the phenomenon is even more evident, with a much stronger signal for the young compared to the older participants. So that's very reasonable. People age and their brain activity declines, their brain functionality declines, which is very, again, coherent and expected, but perhaps even a bit sad, because it would suggest the idea that with aging there is simply a decline in the brain. Fortunately, it's not just about that. At least in healthy aging, there is not just decline in brain activity, but there is also functional reorganization of the brain activity. And this is something that is suggested by what we observed in the left auditory cortex, because here, once again, you got the same colors, same kind of scenarios before, but in this case, the results are different. For the memorized condition, for instance, you can see that the older adults presented the stronger activity in this, especially in this positive component of the signal here. So for all the sounds, essentially, there was this stronger activity for the older adults. And what is interesting is that the stronger activity disappears when we had the variations instead. So here you can still see it for the first sound, because the first sound is the same as the previously memorized. But then for the other sounds, this positive peak disappear, or at least the difference between the young and older adults disappear. Same thing when we introduce the variation at tone number 4. So for the first 3, the difference is there, but for the last 2, the difference essentially disappears. And this is also very coherent with the behavioral results that we could observe in this case, because in the behavioral results, there was no impairment in the ability of older adults to recognize the previously memorized sequences, but there was indeed an impairment in the ability of these older participants to recognize the varied musical sequences. What is interesting is that here, where there is no difference in the behavior for the recognition of the previously memorized musical sequences, there is instead a difference in the brain, because the brain is reshaping its internal balance, with a reduction in the hippocampal regions and in increase, potentially showing compensatory mechanisms in the left auditory cortex. So just to get to some conclusions, here we could show the brain spatiotemporal dynamics of this recognition of musical sequences, highlighting how is important this fast-scale activity and communication between a set of brain regions, which are especially auditory cortices, hippocampus, inferior temporal cortex, anterior cingulate gyrus, and ventromedial prefrontal cortex. And then we could also highlight this reshape in the brain, in the aging brain, happening with this enhanced activity in the left auditory cortex and decreased activity in right hippocampus and inferior temporal cortex. And then again, the same question as before. So why is this relevant? And especially, what can we do next? Or what are we doing right now? So this is very important, because we can use this knowledge to study disorders of memory. For instance, disorders like Alzheimer's disease and dementia caused by Alzheimer's disease. And this is important because the long-term goal here is to then conduct a longitudinal design where we can follow these older adults over the years and hopefully connect their aging trajectory, if in healthy or pathological aging, with the neural data that we will collect, neural and behavioral data. And this is going to be very important because it might allow us to build some screening tools that are based on the combination between these musical paradigms and these state-of-the-art neuroscientific and neurophysiological techniques that could allow us to quantify the risk, the individual risk that these older adults might have of developing dementia. So with that, I thank you very much again for inviting me to give this talk. And I also want to thank all my colleagues, especially in Aarhus and in Oxford. And I also thank all the foundations and institutions that supported and collaborated with us in this research. Finally, if you are interested in this research and you want to read more, here we have the QR codes to 2 of the key papers that we published recently on these topics. Thank you very much.